Hi everyone, my name is Carson Stringer. I'm a group leader at HHMI Genelli Research Campus, and I'm going to tell you today about how to use Cellpose 2.0, a new version of Cellpose that me and Marius Parkitsariou created. And so hopefully with this algorithm, you're going to be able to train your own model on your own data to get high performance cellular segmentation. So the problem we're trying to solve here with Cellpose is cellular segmentation. So you have an image of cells where, for instance, you have maybe two channels where the cytoplasm is in green and the nuclei are labeled in blue. You want to draw circles around each of these cells. And ultimately, what Cellpose and these algorithms return is every pixel is labeled with a number, which corresponds to which cell that pixel is in, and then zeros for the background. And so you might need to do this segmentation for a variety of analyses, like say you want to count cells in your images, you want to monitor cell shape changes over time, you might want to quantify gene expression, for instance with in situ RNA sequencing, or you might want to record neural activity where you're going to be segmenting, for instance, these are neurons recorded with calcium imaging. And so we created Cellpose as a generalist algorithm to try to segment all of these different cell types with the same algorithm. So how did we achieve a generalist algorithm? The first thing we did was we created a large data set with 70,000 cells. Here are example images from our test set. Additionally, we created an auxiliary representation of these ROIs, of these cell masks. So our Cellpose network, which is a UNet architecture, predicts from the image, each of these cells is represented as this flow representation. And so the network is trained to predict these flow representations, and then post hoc, we run dynamics on the flows that the network predicts, and ultimately return masks for the user to use these ROIs for each of the cells. And you can see that Cellpose this single model can do well across a variety of images. So in these test images, which were not used for training, the ground truth labels are in yellow and the Cellpose predicted labels are in red. And you can see that Cellpose is able to segment these wide variety of images. But so now I'm going to show you how to install and run Cellpose in the GUI. And so to do this, we're going to use the first thing I'm assuming here is that you've installed Anaconda Python on your computer, and then we're going to create a new environment to run Cellpose in. And so we can say here, conda create name Cellpose Python equals 3.8. That's the version of Python we're going to use. And this is going to create a new environment for us with just Python 3.8 inside of it. And then we're going to install Cellpose inside of it. And so there shouldn't be any conflicts with any other packages inside this. So I've activated my new Cellpose environment. And now I can say pip install Cellpose GUI. So this will install the GUI dependencies as well. All right, so it's successfully installed everything. And so now we're going to say Python m Cellpose to start Cellpose here. Now we have the Cellpose GUI opened up, and we can drag and drop images, for instance. Here's an example of an image that we can drag in. And we can segment this image with the Cellpose algorithm. We can also draw on, I just right-clicked here to draw an ROI. I can delete ROIs with control click. So let's just run the Cellpose algorithm on this data. We'll use the default cell diameter, but you would set this to the diameter of the cells that you have in your image. You want to make sure this this case, there's just one channel, and so it's gray that we're going to segment, and there's no nuclear channel. And we're going to run the cyto model. And you can always go in the command line, you're going to see what's going on inside the GUI. All right, and you can see here we found 213 ROIs. And you can also then go in and correct these ROIs if you'd like. You can delete some of them, draw new ones, etc. All right, let's drag in an image that has a nuclear channel as well. So here's an image with the cytoplasm in green and the nuclear channel in red. And so in this case, we're going to set the channel to segment to green and the second channel, the nuclear channel, to red. And again, you want to make sure this diameter looks right for your cells. You can also run this calibrate option to find the approximate diameter for the cells in the image, although it won't always be perfect, so it's usually better to use your own. But here we can run the calibrate and see what it suggests as the correct diameter. Okay, it says 37.6. Okay, that looks pretty good. And so now let's run the cyto model, the default model, and Cellpose on this data. And there we go. We have this. Now we found 74 ROIs, and these look pretty good. I can turn on the outlines and turn off the masks. It makes it a little easier to see these things. You can see that we found, looks like we found these ROIs and did not merge any of these ROIs together. And so, and an important thing to note is that the algorithm will be somewhat sensitive to the size, so you want to make sure that's similar to the size of the ROIs in your image. And also, if you do have a nuclear channel, it does help to add it because it gives the network context to know that there's 2 cells here, for instance, rather than 1. So it knows that it needs to put a boundary here wherever, I mean, in particular, where it sees that there's a boundary. Okay, and so now let's go back and see what kind of features we've added. So this is the Cellpose. These are all the features that were available in Cellpose 1.0. You can also view the flow representations of the cell probability map. And so these are all features available in Cellpose 1.0. Let's go into Cellpose 2.0 now. All right, so we were kind of hoping with Cellpose 1.0, we've been asking for user data and we've gotten user data, and we've been trying to make this big generalist model that accounts for all the possible data that people might have. But what we found is that biologists have different needs in mind when they segment their images, and so segmentation styles vary across annotators, across labs, across needs. And so, for instance, in our Cellpose data set, we had specific biases in our segmentation styles, like we only segmented cells which had cytoplasm. We didn't segment cells here. You can see that you only can see the nucleus and you don't see any cytoplasm stain. We also, for instance, if it was ambiguous, we didn't segment these long processes. We also kind of segmented as much as possible for each cell, kind of encompassing all of the cytoplasm possible. And you can see that in this image as well. Whereas other data sets from other labs, such as the TissueNet dataset, which is a large data set of tissue data collected on a variety of platforms, they segmented the nuclei in all of these images, regardless of whether or not there was cytoplasm there. They often didn't segment all of the cytoplasm in ambiguous cases. And so you can see that in here and the ambiguity in this case here. So that's a different segmentation style than the Cellpose model. And so the single model can't say that to segment nuclei in one image and not in another. So that's a potential problem. Similarly, we saw different annotation styles in another large data set that was recently released, the live cell data set, where basically the annotators did not segment in areas of ambiguity, and so there's large areas of the image that might not be segmented depending on the density. And also they were careful to segment all of these processes. So those are kind of 2 sort of different biases that the live cell data set has compared to other data sets. And so we kind of have this problem that a single model cannot segment images in different ways. And so we potentially need multiple models in order to capture the variability across segmentation styles. And so we tested whether or not it helps to have multiple models by first computing segmentation styles of the images. And so the styles that we get are here in the network, basically an average pool of the output of the lowest layer in the network. We use that as our style vector. So it's 256 numbers for each image, which kind of represents overall approximate style of the image. And then we clustered these segmentation styles. And you can see, so CP stands for Cellpose dataset, TN stands for TissueNet dataset, and LC stands for live cell dataset. You can see these 9 different clusters of images that were found using basically using the Leiden algorithm on the segmentation styles that we got out of the Cellpose network. And what we found was that if we use these specific networks, rather than using a single model trained on all of the data, we get an increase in accuracy here. So 0 is that you're the same as a general model that has all the data. And basically for all of these different data types, these 9 different data types, having a special model for each of them was better than using a single general model. And so this suggests that having different models is useful. And we can see even we can use basically we put all of these models into the GUI so that you can take the same image and apply different networks to it and see different styles emerge, different segmentation styles from the same image. You'll get these different segmentations here. And so I can show that now in the GUI. If we go back to the GUI here, we have this image loaded in. This is the segmentation from our default Cyto model from Cellpose. It's going to be similar to, this is the CP model, which was trained on the subset of the Cellpose dataset, which has this data. So this is the CP model here. You can see it segments all of these processes. Now we can try applying, for instance, one of these TissueNet models. And you can see that it basically largely segments things close to the nuclei. It doesn't segment all of the cytoplasm here. And so we have these, now basically we have all of these different models in the GUI that a user can click on easily to run on their data and decide which one works best. And so now we have all of these models. We kind of, does this mean that like for every single biologist, for every problem, we're going to have to make all of these different models? And do we need lots of data? So like the TissueNet dataset and the live cell dataset were hundreds and thousands of cells. So we kind of have this concern that maybe everyone needs lots of data to make their own models, to have their own little buttons inside Cellpose. But we don't think that's the case. We think that models with limited data can perform well, particularly if you started a good pre-trained model like Cellpose. So hopefully, for most people, you can use a limited number of annotations and have a good model of your data, starting from one of these models from our model zoo that I showed you. Okay, so here's basically how well a model does with limited data. So we start with the TissueNet breast spectra class, and we apply the Cellpose model to it. We get an accuracy of 0.36. And we can start adding training ROIs and retraining the Cellpose model with the segmentation. With this style in particular, you can see where the nuclei that weren't segmented here, they're now being segmented here when 400 training ROIs are added. If we add almost 2,000 training ROIs, you can see that the accuracy goes up even more. But as we add more training ROIs, the accuracy appears to saturate. And we can quantify that across all test images and all data types. What we find is that the accuracy increases as a function of the number of training ROIs, but starts to saturate. And the solid line is starting from the pre-trained Cellpose model versus starting from scratch, which is the dashed line. And the solid line here is the Mesmer model which is the state-of-the-art model that was trained on all of the images in the TissueNet dataset. And so already with only 1,000 ROIs, starting from our pre-trained Cellpose model, we can outperform this state-of-the-art model trained on all of the data. So this suggests that maybe you don't need hundreds of thousands of ROIs in order to create a good model for your data if you start from a decent pre-trained model. We saw this hold true for the live cell data set as well. And so our starting from the pre-trained model, you can see is even more important in the live cell data set. You can see this curve is much higher than the starting from scratch curve. And it saturates relatively quickly around 1,000 to 2,000 ROIs. And so this suggests that we don't need a lot of data to train our models. And we further think you need you could use even less data if you use a human in the loop approach, which we've implemented in the Cellpose GUI, which is where you run Cellpose 1.0 on your image, and you have this automatic segmentation from the Cellpose model. You correct this segmentation, and then you add this image to the training set. You retrain Cellpose, and you're hopefully going to have a better segmentation the next time, and you'll have to do less manual curation. So here's an example of going through the human in the loop procedure with the TissueNet dataset. And so on the first image, we run Cellpose 1.0, and 82 out of the 113 ROIs need to be corrected. But on the second time, after the model's been trained with just this single image, we see that only 32 ROIs need to be added out of 174. And then continuing this procedure, only tens of ROIs need to be added rather than hundreds in order to correct these segmentations. And so here's how this works inside the GUI. So you saw how we can run the model and inside the GUI. And now here we are correcting the segmentation. So that was an error there that from the original Cellpose model. There's an error being corrected. All right, and so once all of the corrections are performed by the user can go up to this models file menu up here and start training a new model. So here the segmentation looks good. So we can train a new model on the single image. Okay, and so now the training is running. And so this will be relatively fast if you have a GPU in your computer. So in this case, it takes around 30 seconds to train the model. And so now the model was automatically applied to the next image in the folder, and you could already see that it was doing fairly well. We can move on to 5 images later as the users continuously updated the model and updated the segmentations. We're already at a place where the segmentation looks pretty good. Oh, another new feature as well in Cellpose 2.0 is you can just click the R and the G or the B buttons to flash on and off different channels, which might help you to see things. You can also flash on and off the outlines, as you can see being done here with the Z button. All right, so now the model is being trained on 5 images. And now it was applied to the next image in the folder, and you can see it did a very good job of segmenting it. And we can drag and drop in other images here. So here's one of the images that wasn't used. You can see the model was added to the GUI, the one that you just trained. And that looks pretty good. You can drag and drop in another image and again run the model, which this will stay in the GUI for you. You can also remove it if you don't want it there. And it's also saved in the original folder where you've segmented, where you've been doing this segmentation of the images, whatever working folder you're working in. Okay, so what we found in this human in the loop performance was that doing human in the loop might only require 167 manual ROIs to be corrected or drawn, whereas if you do the manual annotation from scratch, it requires many more ROIs, like around 663 to obtain to segment all 5 of these images that I was showing you from human in the loop. And so, and this is always starting from the Cellpose model. If we start from scratch and do it online, we require more ROIs as well, around 370. This is still less than doing it offline from scratch, but it's still a lot more than, it's 2 times more than doing it from the Cellpose model. And what you'll see is that, so basically as you have more training images, you can see we need the fewest number of manual ROIs drawn for when we do it online, starting from the Cellpose model. That's what this graph shows. Less than with online scratch, and then these 2 curves are the offline training where you have to draw all the ROIs. And what we found was that the accuracy was similar for the model trained in the loop versus offline. So the online offline curves are the blue and the orange curves here respectively, and so the accuracy is very similar, and it gets up to basically the maximum performance, which is within human agreement, where the user segmented the same image twice and compared how similar the segmentations were between those 2 images, those 2 segmentations. Whereas you can see the scratch model required more ROIs in the loop and also still performed worse after 5 images. So generally speaking, if your data is somewhat close to the Cellpose dataset, it should generally work better to start with the Cellpose dataset, and it should generally require less data to use the Cellpose model in the loop in the GUI. And so I'm not going to, I've shown you how things work in the GUI here in these videos. We have more videos on the GitHub. And now I'm going to show you also you can train the model inside a Google Colab notebook. So if you don't have access to a GPU on your local computer, you can use Google Colab. And so here's a notebook that's also linked inside the Cellpose GitHub. So we're going to run Cellpose 2.0 here, and just to note the things that we were doing, your custom model, after you've trained it, will show up here after you've run this training procedure here. And also there's training instructions here for you inside the GUI to help you go through it. And so once you've run the training here, you might want to run your model on many images, maybe using a GPU, where you will want to do it on Google Colab. Or if you don't have a local GPU, you might have to do all the manual annotations locally and then upload them to Google Colab to make them run faster. Or you can also, if you have a decent CPU, you can wait 5 to 10 minutes for the model to train rather than a minute. Okay, so now we're going to run Cellpose 2.0 with a GPU. So inside this notebook, we'll show you how to load a custom model and run this model on your images with the GPU in Google Colab. We'll also show how you can train a custom model using your labels, so the seg.mpy files, which are saved inside the GUI. And so you can see, for instance, when I click here, that there's these images that have seg.mpy files show up to use for training, and inside my folder here, anytime I run the algorithm and save the segmentations with Control S, I get a seg.mpy file which contains the masks. And so that's the default kind of saving output from the GUI. You can also save other types of masks from the GUI. You can save masks as PNGs, for example, or you can save outlines for image J. But you can use these seg.mpy files to retrain your model, and that's what's used in this human in the loop in this training procedure. It looks for all the seg.mpy files in the folder, and for instance, it finds this one here we were talking about with 213 masks. Okay, so let's say you've taken all your images and all your seg.mpy files and put them in a Google Drive folder, and we're going to now train that model on the GPU in Google Colab. And so this is saying you can mount your Google Drive here to do this. Okay, so the first step here is to install Cellpose in this Google Colab environment. And then we're going to check that the GPU is working. So this NVIDIA SME command is going to show us what GPU we have and the GPU usage. So we have a Tesla K80 here. And then we're importing Cellpose and checking if the Cellpose library sees the GPU. So that's a core.use GPU function here. And it says yes. Okay, so everything imported correctly. And so this next step here is if you're going to mount your Google Drive, this is where you would do it. You would run this cell and put in your credentials for Google Drive to link it. And then it would show up in this file folder here. But for now, we're going to use sample example data that I'm going to download next. And so if you want to just try out this notebook, you can download our sample data here. And also this sidebar is nice too if you want to see all of the table of contents for what's going on inside the notebook. Okay, and so we downloaded this zip file and this human in the loop folder, which has two folders. It has our training images, which we have our seg.mpy files for. And it also has our test images, which again, we have the seg.mpy files for, which are our manual annotations again. Okay, and so now let's just see what the training images look like and their labels. All right, and so we have the image here. So you can see that the cytoplasm is in green and the nuclei are in red. And each of these images here are the images where the background is 0 and each of the masks is 1, 2, 3, 4, and so on. And so that's kind of what these colors look like. And this was done as human in the loop. And so you might see this color gradient, which is just like, that's what happens when you run Cellpose. The cells will be kind of in order. You can see actually the corrections are kind of these yellow ones that were added at the end by the user in the human in the loop. So you can see which kind of corrections were done here. But anyway, that doesn't matter so much here. We're just going to train with all of this data, whether or not it was collected with human in the loop or just drawn completely from scratch. Okay. All right, so now we're going to train the model, and you could skip this step if you already have a pre-trained model that you're going to upload to Google Drive and mount here. So what we need for training the model is we need the training directory and the test directory. So these are the images. These are these folders here, which have your images and your seg.mpy files. You don't necessarily need a test directory, but you can have one to allow you to see how test performance looks. And then there's a few other training parameters. One of the main ones here is the number of epochs. So we run usually with 100 epochs, but you could run with more, particularly if you have a lot of training images. Oh, and another thing is you want to choose what model to start from. And so again, we have this model zoo with lots of different models, which we were showing here. These 9 different models plus our bigger models trained on more data. And so you can choose that model to start with, and then you're going to say the name of the model you're going to save. And so our paths to our images and masks. So we have this human in the loop folder. We have the training folder, the test folder, which we put in here. We're going to start with the cyto model, but again, we could choose different models. We're going to call the model CP TissueNet. We're going to train for 100 epochs. We're going to choose the channel for training to be green because the cytoplasm was in green. And then the second training channel is for nuclei, and the nuclei were in red. So we're going to choose that. And then we have the learning rate and the weight decay here, which we recommend keeping fixed. And so we'll run this to set all these parameters in the background. And then we're going to use these parameters to train the model. And also we have a little section here, which basically takes all these parameters you put in here and converts it to what it would look like if you were going to run this on the command line. So you would have to change your paths because these directories will be different on your local computer, but this gives you an idea of what the command would be to run your model on the command line, like how you would put in your channels according to what you did up there, the number of epochs, the options, etc. And so this might be helpful for you if you want to go back and train locally as well if you have a GPU in your computer. Okay, so now let's train the model. So here's how to train the model using in a Jupyter notebook what the script looks like. So the first thing, so we're going to start the logger, which is just going to tell us what's going on inside the code as things run. And then we're going to declare the model. And so there's this models.cellpose model here. If you've used Cellpose in the past, you might have used just Cellpose without Cellpose model here. And that Cellpose without the model is the version that has both the size model and the Cellpose model inside of it. But here we don't have a size model. We're just going to train a Cellpose model just with your images. We're not going to train this automated size model. And so the two main things we need here, we're going to say that we're going to use the GPU. This will be true. And then our initial model, which we set above, is this string Cyto here. And so those two arguments go in to define our model. Then we're going to set our channels, which were defined above. So green is 2 and red is 1, RGB. And then we're going to get our files from our directories. And so this is an internal function in Cellpose that allows you to get all the images and the corresponding seg files from a training directory and a test directory. And so this gives us these outputs here. And then we're going to train our model. And so we're going to put in the train data, our training labels, our test data, our test labels, and then the channels, the save path, the number of epochs, learning rate, weight decay, and number of image per epoch, which we'll keep fixed to 8, and then the model name, which you declared above as CP TissueNet. And then we're going to run this. So the first thing it's doing is it's actually downloading the Cellpose Cyto model so that it can use it. It's going to generate the flows, these flow representations for the cells that we need to use for training. So that's what's happening here. It's going to compute the mean of the training label mask diameters. So it found that the masks on average had a diameter of 19. And that's actually going to be saved inside the model. And so if you run the model with diameter equals 0 in the future, it's going to just default to this 19 value. So in the past, if you had a diameter of 0, if you set the diameter to 0, it would run the size, the automated size model, but we don't have that here. And so it's just going to set it to the size of your training images. And so you can see it's training the network. It's going to set the learning rate, the weight decay, and so on here. And you can see the number of images and the number of images per epoch. All right, and now it's finished, and you saw it took a little over a minute, not too long, on these 5 training images. And now the next thing we can do is see how well our model did. And so we're going to look at our test images. And so we're going to grab our test files from our tester, which has the test data and the test labels. Again, you could set these by hand, but we're using our internal function to get these, to load these in for us. And then we're going to run, now we're going to run this model.eval function. So this is a method of this Cellpose model class. So model.eval is a function which we can use to call on our data to run the model. And you can see if I mouse over this in Google Colab, it gives you all of these options. So there's lots of options here because this is basically the main function in Cellpose, which you can even say if you have a 3D image, you're going to run with 3D. There's lots of options here, and you can see more information about the options if you, if you scroll down here, and you can also see more about these options in the read the docs for Cellpose on this that are linked on the Cellpose GitHub. Okay, and you can see it returns a few things. It returns the masks, the flows, which are those flow representations we saw of the cells, and also the style vectors. And in this case, we just need the masks, so I'm just actually going to take the first output of this function, which are the masks here. And so the main things we need are the channels. So we're going to set the channels to chan and chan2, which were 2 and 1 for these types of images. And I'm going to set the diameter here. I'm going to set it specifically to diam labels, which is up here, which is the average diameter of the training labels. But again, I could set that to 0 and it would just use that automatically. All right. And so what it found was that it got an average precision at an IOU threshold of 0.5 of 0.693. And so already we're at a relatively high accuracy with just 5 training images. And this is another function here you can use inside Cellpose, which is the average precision function, which tells you this accuracy for each of the images. Okay, and we can plot how good these masks look. You can see the original, test image here, the predicted labels from the model, and the true labels. And so you can see, like, for instance, the model learned this bias to segment nuclei without cytoplasm, which the original Cellpose model doesn't have that bias. And you can see it segmented these here. Okay, and so we've gone through the whole training model section. And so now we're going to look at using your custom model, which you might have trained already in the GUI inside this Colab notebook, or if you were ultimately going to run it in your own Jupyter notebook or script. So we need so we're going to take the custom model trained from above, which if we go here, and this is the case on your local computer too, whatever folder I used for my training data, that's where the model is going to be saved. So you can see it's saved here. So this is where my model lives. If you're using it inside the GUI, the model is also saved to your hidden folder .cellpose, which is why you can always see it there to use. But there will always be then 2 copies, one that's always accessible to the GUI, and then this one in your training folder here. And so we're going to set our model path to this path here, human in the loop /train /model /CP TissueNet. Then our path to our images, which we're going to use our test folder. And then I'm going to use green again as our cytoplasm channel and red as our nuclear channel. I'm going to set to 0 to use the diameter from the training set, but I could use a different diameter. And then there's these other options, the flow threshold and subflow threshold, which are explained more on the read the docs as well, but these are options you can set here too. So one small thing I did here was I moved the test images. Oh, oops, I forgot to run this. Sorry. So you run this form here to save all of these options to variables. And then what I did here was I moved these files to a new folder here, this eval folder, just so I don't overwrite these manual segmentations that we had from here before with the automated segmentations from the algorithm. Okay, and so if I was going to run this on my local computer, here's what the command line command would look like. And so again, you can copy paste this to your local computer if you want to, or you can run it inside a Jupyter notebook or in a script. And so this is how you would do it. Again, I'm using one of these internal functions just to get all the image files in the folder, ignoring any _masks files, which you might have. And then I load in all of the images here with our built-in function imread. And then again, we're going to declare the model. And so here we're using the Cellpose model again. We're going to set GPU equals true. And we're going to set now instead of using a model type, which is what we used for those built-in models, we're going to use pre-trained model, and we're going to put as the pre-trained model the model path. And so you can see what this is set to. Oops, let me mouse here. Oh, maybe it was better here. It's going to be set to this model path we have, human in the loop / train / model / CP tissue net. And so we're going to give it that path and it's going to load in the model. And inside the model, it's going to load in the model has the diameter of the training labels there. So that's going to be loaded in as well and be set to model.diam labels. And here what we're doing is we're taking this diameter, if the diameter equals 0, otherwise we're taking the diameter that you've set above here. Okay. And then we're going to run this model.eval again, and we're going to output the masks, flows, and styles. So we input the images, the channels, the diameter, and then the flow threshold and cell prop threshold that you put in. All right, and so this ran, and so now we can save the outputs in various ways. You can save the outputs to these _seg MPY files that you can load into our GUI. You can also save the outputs as TIFFs or PNGs or text files for ImageJ. And so here I'm basically setting most of these things true here. So like if you want to save the masks as PNGs, if you want to save the masks as TIFFs, which is the only way, for instance, you can save 3D masks, you can't save 3D PNGs. Or if you want to save the text outlines for ImageJ. And so we can run that here. And you're going to see all of these things showing up here. And so if I press reload here, there's a lot of stuff here now because we have the seg.mpy file for each file, and then we have the outlines and the masks as PNGs and as TIFF. And we also have this cp output.png, which I'll show here, which kind of saves as a nice way for you to view your data if you'd like to. So you can see the image in grayscale with the predicted outlines, the predicted masks, and then also the Cellpose here. And so that's it for this notebook, and this notebook is available in the Cellpose GitHub for you to try out and to modify to however works best for your workflow. Okay, so in summary, we found that there are various segmentation styles that people use. So we've created this model zoo inside Cellpose for the user to be able to quickly access. Again, all of these are sitting here for you to easily be able to just click on to run a new model on your data. And so then if you have a good pre-trained model, you don't need a lot of data to create a new model. So hopefully you can find a model inside the model zoo, which is okay for your data, and start there and start training with your own data. We found that training models is faster if you use human in the loop training. So if you're able to have a local GPU and use the GUI to segment the images and in the loop add these images to your training set and re-segment the images, then things will go faster. If not, you can always segment them manually, all of the images, and then upload them to Google Drive as I was showing you, and use our Google Colab notebook. So in the future, we're considering we might host new user models. For now, we're still asking users to submit segmentations through the GUI. And so that's this option here. You can send your manually labeled data to our server. And we actually do have a model here, this Cyto2 model, which you can run, which has a few hundreds of images that users have sent to us in the past. And so I'd like to thank again my collaborator, Marius Pakitsariou, and my funding from HHMI Genelia Research Campus.